and its applications in drug discovery. Today, our first lecture is by Dr. Pratik Chatterjee. Dr. Pratik Chatterjee is an associate researcher skilled in biophysics, physical chemistry, material science, nanomaterial spectroscopy, computational tools and languages, as well as synthesis and characterization. He gained his postdoctoral research experience in biophysics at SMU, Seoul, South Korea. Sir, would you mind taking over? Sir?
Yes. Thank you. Can you hear me? Hello.
Sir.
Yes. Yeah. Thank you very much for your kind introduction. So, shall I share the screen of my presentation slides?
Of course, sir. Yes.
Yeah. Okay. Is it visible right now?
Yes, sir.
Okay. So, is it full screen?
Yes, sir.
Yeah. So, shall I start over?
Yes, sir. Absolutely.
Okay. Thank you very much. And good morning, everybody. I thank all the organizers and the esteemed committee members present in this particular symposium, in arranging this particular symposium, for giving me this kind opportunity to present a bit of theory and application of molecular dynamic simulations, which will be relevant for protein folding and drug discovery prospects. So, even though some concepts of protein folding, molecular dynamics, and drug discovery will be familiar with the audience who will be present here, but in order to connect the dots in between the concepts, so I may have some generic, I may give some generic idea of these basic concepts, and please bear with me if you know the trivial things already. So initially, the outline of I present you the outline of protein folding, and for that I give you the synthetic pathway of protein folding, which is like proteins are the building blocks of living organisms, which is consisting of amino acids, and biosynthesis of proteins happen in the ribosome in presence of translational RNA, and the sequence specificity of proteins is encoded in DNA, and the corresponding message is translated by the messenger RNA. And likewise, the protein is synthesized within the cells in the ribosome, and there are different pathways of protein folding, and the proteins which are folded get globular structures in due course of time, and the misfolded proteins go enzymatic degradation and get excreted out. Now, the proteome, which is equal to a term called genome, is actually the total number of protein present in a physiological environment. It can change with time, dependent on several factors, responsible for different functional properties of living systems. There are different kind of functionalized proteins which you will be well aware of. So I am not dealing with greater detail here. And the biological activities which are governed by the proteins are like signal transmission, vesicular trafficking, enzymatic reactions, transport, and cellular development. Now, types of protein, especially for MD simulation purpose, we need to know what kind of proteins we are dealing with in the protein folding community, which you will be well aware of. But since it is a theoretical symposium, I give you a few of the examples of these different types of proteins. Like the globular protein can be the chicken villine headpiece 36, which is the smallest model globular protein, which is used in MD simulation studies for comparative purpose, which is actually a helical protein. And on the contrary, there is another globular protein called the WW domain, which is again rich in the beta sheet structure. Now, enzymatic proteins like amylin or islet amyloid polypeptide I am actually giving, because this particular protein, when gets disordered and gets aggregated, is responsible for type 2 diabetes. So this is also then coming under the intrinsically disordered protein, regarding which I will explain to you in a bit more detail in later point. And then transmembrane proteins are like amyloid precursor proteins. If you are well aware of the neurodegenerative diseases like Alzheimer's and Parkinson's and etc., which happen due to the killing of the brain cells due to aggregation of certain peptides. So predominant of those peptides are the amyloid beta peptides, which happen to be a transmembrane peptide, and the mother protein is this particular amyloid precursor protein present in the amyloid precursor protein gamma secretase complex. Now, due to enzymatic cleavage, these particular domain amyloid beta peptide is actually released into the cytosol and then gets disordered, and then is responsible for further neurodegenerative actions. And then chaperones, and this particular, why I have actually classified peptides is because all peptides are proteins, but all proteins are not peptides. So a protein can constitute of different units of several units of peptides, homo or heteropeptides. And so peptides are more or less in general around 50 amino acids long. And so this amyloid beta peptide, which I have mentioned about arising from the amyloid precursor protein, it is considered to be a peptide in nature, because anyways it is a fragment of a mother protein. So just for a nomenclature purpose, I categorize this particular term as well. Now, if we deal with the concept of protein folding, although I know that in the undergraduate classes thermodynamics has been taught, I will actually give a brief briefing of the thermodynamic equations. The first and foremost is the H is equal to E plus PV. H is the enthalpy or the energy of the system, and E is the corresponding potential energy or the internal energy, and the PV is the kinetic energy or the work done by the system. Now, for a protein folding process, this PV term is too small. So the difference which is coming is actually due to the change in the internal energy, and therefore the enthalpy of the protein can be generically equivalent to the internal energy or E of the protein, which can be mentioned as the EU or the protein internal energy in due course of time in different in different lectures or in different sources. Now, this is the enthalpy, the energetic term, and then there is a free energetic term, the G, Gibbs free energy, which actually depends on the enthalpic term as well as the entropic term. The entropic term is also dependent on temperature, so it's a negative term. So more the entropy, the more negative and thereby the more stable will be the Gibbs free energy. I mean, more negative will be the Gibbs free energy, and the more stable will be the system, biomolecular system or the protein. And with respect to the protein folding process, there is another additional term called F, or the effective energy, which is a summation of the EU, which is the protein internal energy or the enthalpic term, plus the solvation free energy. This summed up term is actually the effective energy, which actually drives the energetics to the more stabler conformation. So these outlines I have given, and I will discuss generically regarding the protein folding phenomenon, considering these particular terms. Now, we already know that protein folding process is the process of attaining the three-dimensional structure of protein, and the timescales of protein folding is actually varying from microseconds to minutes. Three-dimensional, as we know, we have discussed previously, the three-dimensional structural representation of protein is actually embedded in its sequence. And conformational states with higher stability have lesser number of conformations, which is entropically unfavorable. So if there is more entropy, the system will be more disordered. But as we can see here from the pictorial representation, that the protein is actually more or less linear or a compact-like structure without any secondary structure. But in due course of time, it attains metastable states and then gets to the global minimum, happening to have more structural propensity and the globular compactness. So that means it happens with the decrease in entropy. So if there is a decrease in entropy, so negative will be positive, and so the free energetic term will actually increase because of decrease in entropy. Even then, if it is attaining the global minimum, which means the free energetic terms is then considerably negative, which means it is actually an enthalpically driven process. Then enthalpy is so dominating, the energy is so stabilizing because of this particular globular compaction or globular conformational propensity, which is undertaken by the protein in the course of the protein folding process, that it stabilizes the the protein folding phenomenon. So entropically unfavorable, as I have discussed, and this conformationally folded states, the global minimum and some other metastable local minimum from which it actually attains the global minimum, are actually distinguished by thermal barriers. So I mean, after it attains the global conformation, at ambient condition, it happens to be, I mean, the protein will not get back to its initial metastable conformation because of this thermal barrier present in between these two conformational states. Different techniques have again actually focused this to be an enthalpy driven process, and we are actually having more and more knowledge in terms of protein folding because because of the growing protein folding community and the more elucidation of folded protein structures of more than 100,000 present in the RCSB database. Now, if we want to know the concept of protein folding in general, I will briefly explain the Levinthal's paradox, which was actually claimed by Cyrus Levinthal in 1969. Sorry. So if a standard amino acid chain with specific stable conformations samples all possible microstates which it can ideally attain, the time taken to sample the corresponding folding pathway may be equal to or even more than the age of the universe. So what he actually meant is actually described in this particular illustration. Suppose an amino acid chain of 100 residues, which will have corresponding 99 peptide bonds, and therefore each peptide bond has two phi-side Ramachandran dihedral angles, as you might all know. So there will be 198 phi-side dihedral angles. So as we see here, that the structural features which are obtained generically from experiments like X-ray or NMR, those are, I mean, the ultimate global conformation is not mapped, but there an average conformation based on 3 or 4 globular ensembler structures are represented by NMR or XS or some other techniques. So if there are 4 identified stable conformations, so there are 198 phi-side dihedral angles in Ramachandran's case. So if it attains, I mean, if it has to attain all the distributions of the phi-side dihedral angles, until and unless it attains its global conformational propensity with definitive phi-side dihedral angles present in its ultimate conformation, the total number of conformations to be attained will be around 4 to the power 198, which will take a longer time than the age of the universe to attain this global minimum conformational ensemble. Therefore, this suggests that there are selected finite pathway in protein folding, and so protein folding phenomenon is attained by selected intermediate states. Now, the evolution of protein folding mechanism pathway actually varied with time. Initially, it was, I mean, there were preliminary concepts of ion paired hydrogen bonding model, like which happens due to interaction like hydrogen bonding like interactions. between the polar moieties of the protein, but the explanation behind that was it would happen by decrease in volume. But on the contrary, protein folding increases in volume. And then there are other models like one-dimensionalizing model or polyelectrolyte model, like which will actually say that the conformational propensity is dependent on the salt concentration, more the polarity of the environment. Therefore, the greater the shielding power between two polar states present, in the protein, and therefore the attraction or repulsion, whichever is responsible for driving the protein folding, will be less, and therefore the protein folding will be denatured in presence of higher polarity medium. Recent models which have been actually discussed are framework model, where a protein is believed to be in its nascent state, actually as described in the folding funnel. It believes that in the nascent state, it attains the secondary structural propensity and followed by globular compaction by forming collapsed globular structure. And the more prevalent in today's times is the hydrophobic collapse model, where protein in its nascent state forms molten globule state followed by development of secondary structures. So immediately after its generation from the ribosome, it attains a molten globule kind of metastable state, which is actually like a hydrophobic collapse kind of thing, which means the hydrophobic moieties of the protein come closer to each other, and then it forms a globular-like structure before the formation of its corresponding secondary structure. In terms of this, I just present recent data of the EU, GSOLV, and F I had actually defined in the protein folding while discussing the protein folding phenomenon. This is the effective energy which is actually driving the negative energy of protein folding, or the stability of the protein folding, which is a summation of the protein internal energy, or the enthalpy, as well as a solvation free energy. So we see for a particular intrinsically disordered protein, from my case study, is that these are, the blue and red are the charged residues, happening to be the negative and the positively charged residues. So obviously the electricity, it seems that the electrostatic interaction between the charged residues is actually coming into play in the course of the ensemble of structure generated by the simulations. But we see that both the internal energy as well as the solvation free energy is actually governed by the are regulated more by the charge residues if we see the standard deviation of these EU and GSOLV parameters. But it is intuitive enough that if a protein is stable on its own, then it will not interact more with the solvent and vice versa. So since this EU and GSOLV, therefore, the internal energy and the solvation energy are therefore anti-correlated with each other. Therefore, the effective energy actually detects that the contribution of the charged residues, which are so magnified in the individual energetic and the free energetic solvation free energetic term, is actually cancelling out. And then if we consider the non-charged residues contribution, it will be more or less the same as that of the charged residues, in qualitative support of the concept of the hydrophobic collapse model. Now, just because we are dealing with the protein folding phenomenon, we will also discuss the intrinsically disordered proteins energy landscape. So this is the globular structure of the folding funnel of a globular protein, but each of the globular proteins have a more negative steepened funnel, which is actually its amyloidogenic state, which if it happens to be, or if it happens to get out of this thermal barrier, as I discussed before, and can attain this kind of structures, it can maybe form amyloid-like fibrils. Like sickle cell anemia is formed by the amyloid-like fibers formed from the misfolding of hemoglobin. Hemoglobin is a globular protein, but most of the globular proteins have a definitive folding funnel. But on the contrary, as I had discussed before, in terms of the amyloid beta peptide coming from the intrinsically, I mean, from the amyloid precursor protein, there is a special categorization of proteins called the intrinsically disordered proteins or IDPs, whose energy landscape is very rugged in normal, normal ambient condition. It is very shallow, so it cannot have a stable minimum in ambient conditions, and therefore it has a tendency to self assemble with each other, forming aggregates, oligomers, and fibrils. And we see that these oligomers and fibrils are believed to be responsible for several neurodegenerative and other diseases. So this is again a schematics of the protein folding phenomenon and how the reversible reaction mechanism of intermediate formation actually forms the prefibrillar species and then leading to the amyloid fibril formation. Now, if we are discussing about the protein folding, the solvent has a greater influence in driving the protein folding phenomenon. So biological solvents are actually ubiquitously surrounded by aqueous environment, which effectively tunes the formous properties. And water properties are actually considerably different in the vicinity of biological molecules to that of the bulk water. Large-scale motions of proteins are coupled to small-scale amplitude of functional groups. So the translational or rotational motion of, the global translational and rotational motion of the protein are actually controlled by the small-scale motions of the proteins as well, which is again fine-tuned by the solvent environment because of its interaction with the side groups, the methyl groups, or the peptide groups, etc. And therefore, change in polarity will alter the protein conformation. And interestingly, if you see the recent studies in experimental and computational aspects of protein folding, up to a certain extent, if we decrease the polarity or increase the temperature, there will be a certain kind of rigidification of the protein structure. That means the protein will try to get rigidified or try to stabilize its inherent stabilizing bonds, hydrogen bonds or other polar bonds in its own, until and unless the breakpoint, energetic breakpoint comes, at which the protein in presence of more energy or less polarity gets degraded. So because of the solvent influence and the importance of water in protein folding, a term biological water is has been coined, and there are eminent researchers all across the globe, even in India, working on this particular field. But again, as we had discussed, that the essential role of hydration water layer is still a matter of debate, because as we know, and we had discussed twice before, that the inherent globular propensity, or what kind of structural or functional pattern it will happen, is encoded in its sequences, and therefore mutational studies actually undertake to see the differences in structural and functional parameters. So therefore, what the exact role of this hydration water layer or the water layer is, whether it is a secondary effect in tuning the overall structure of the protein, and in defining the same, an eminent biophysicist, Pavel Jungwirth, in 2016 actually gave a very good cartoon representation, which is like, if you pull out the fish from water, obviously it will die, it will not live. But if you put in water, it will be guided by water. Its oscillatory movements are actually, not regulated by water, but are helped in undertaking by the surrounding water. But the movement of the fish will actually be dependent on the fish itself. But since there are results going on, there are recent studies which have actually shown that water properties 50 angstrom apart from the surface of the protein is showing different properties than the bulk behavior. So the studies are still going on, and it may take some time more in the decades to come to convert the idea or the hypothesis regarding the same. Now, before starting the molecular dynamic simulations, I focus on the scope of computations in atomic level understanding. Computational methods are an indispensable tool for analyzing complex biomolecular systems. And more accurate calculations are quantum mechanical methodologies like density functional theory. It can elucidate properties of such systems unlimited by system size. And there are some semi-accurate molecular mechanical models like Born-Oppenheimer approximation, which is elucidating the atomistic properties by considering each atoms as charged or uncharged spheres, or considering only the nuclear motion and ignoring the electronic motion or the electronic surroundings. Although this above model is incapable of capturing electronic information, but it is of immense value in understanding larger systems. But again, the time constant is there. Now, atomistic molecular dynamic simulations is a unique technique. It is totally ignoring the partial charges, the polarization or other electronic properties or the nuclear properties of the system, and considering each atom as hard spheres with specific partial charges, and therefore it actually implements Newtonian motion for elucidating structural and dynamical properties. Now, MD studies are actually bridge between the microscopic events and the macroscopic observables, and with the MD studies we can actually have detailed detailed length and time scale studies of different biomolecular ensembles. And the applications of MD studies are on protein folding, lipid, nucleic acids, protein ligand interactions, which will be discussed in a greater detail later, atmospheric science, material science, polymers, etc. So this is the time scale and the length scale of different molecular mechanics or atomistic quantum mechanical scales. Quantum mechanics happens to be within the picosecond time scale. Atomistic scale happens within the nanosecond time scale. A mesoscale, which is like atomistic scale, is each hard sphere, and then mesoscale is again a collection of atoms like the methyl group to be one particular hard sphere, like that. So it is coarse graining is coming in the mesoscale, and then microscale. In the microscale, suppose each protein may be considered as a hard sphere, and then maybe at least in the biological problems, macroscale may be defined as each cell may be a particular hard sphere. So this kind of microscale or macroscale kind of studies are actually undertaken by eminent groups in Jawaharlal Nehru University. But there are in between the fine lines of quantum mechanics and molecular mechanics, there is also a specific technique called quantum mechanical molecular mechanics, QMMM, where the active site is actually calculated quantum mechanically because it may undertake chemical changes. Now with MD simulations, we can only have physical changes. We can only study the physical variation of the ensemble. But if there are changes in the partial electronic charge of the particular ligand or particular molecule within the active site or the frame of reference, then QMMM studies are much better. And then MD simulations with polarizable force fields as well for those kind of studies, and ab initio MD as well, where the electronic configuration of each of the atoms is actually calculated at every step, and the force field or the energetic or the potential energy is reparameterized after every step. And as you can understand, therefore these processes are more computationally costly. So the computational methodology of molecular dynamics is discussed in brief here. So based on the potential energy, inherent potential energy already defined for for the molecular and mechanical systems of interest, biomolecular systems. So we can get the force, the force by the negative gradient of the potential energy. I am just giving a brief introduction of these things here. You can have a detailed understanding with NPTEL lectures, or you might have undertaken these in your undergraduation levels. Now, from the force, we can calculate the mass, velocity, and acceleration, and again the velocity and the position at each corresponding time step, which happens to be the del T happens to be around of 10 to the power minus 15 second for molecular dynamics, is actually computed from the velocity Verlet algorithm. Now, what defines this particular interatomic potential for a particular biomolecular system? Those are, the components of this force field or the potential functions are the bonded terms, which are the bond, angle, and dihedral potentials. So bond potential is also, a spring constant type of potential. You can see by the expression. Similarly, there is the angle, and the dihedral has two kinds of. These are the bonding potentials. Two kinds. One is the proper torsional potential, where the phi is the angle between this particular bond and the plane between the remaining three atoms. And if the four atoms is not sequentially connected, then the angle forming with this, between this particular bond and the remaining three bonds is actually the improper torsion, which is again, denoted by spring constant kind of potential. Now, the non-bonded potentials actually include the Lennard-Jones as well as the electrostatic terms. The Lennard-Jones have a slightly decaying, marginally decaying attractive term, and significantly increasing repulsive term at the point of the armine, where the interaction between the two pairs of atoms is zero. And the electrostatic terms is actually the Coulomb potential between the two charges. It can be plus, minus, minus, plus, and the interaction, whether positive or negative, will actually exponentially decrease because it is a inverse function. The functional is dependent on the inverse of r, so it will exponentially decrease with time. And for computational ease in MD simulations, after 10 or 12 angstrom, it depends on different force fields or packages, this has been artificially made to 0. After a switching distance, suppose 9 or 10 angstrom, this will be parameterized in such a way that at 12 angstrom, the interaction at and greater than 12 angstrom, the interaction between two pairs of atoms will be 0. Now, I just give here, I am not discussing what the different force fields of different community are and the corresponding Wharton model, and how successful they were in actually generating a folded conformation out of an unfolded conformation of Ciccarelli net freeze, as I have told, and WW domain. And you can have a detailed look later on. I have given the reference here. And what is the concept of mirror image convention in simulations? And so for that, we need to understand the periodic boundary conditions. For the periodic boundary condition is that, suppose, this is a two-dimensional representation of a particular cell where different molecules are present. But if we consider only this particular, particular cell, then the number of atoms present in its corresponding surface will be around 1/3 of the total number of atoms, and its corresponding properties will differ than that which is present in the core of the cell. So to avoid that, an algorithm called the periodic boundary condition has been actually defined, which is that there will be equal replicas of same cell in each of the dimensions, and therefore in such a way that if a molecule is actually exiting the cell, a similar mirror image of this molecule is actually entering the cell from the opposite direction. And therefore, the calculation of electrostatics will be in such a way that suppose there, I mean, we want to calculate the electrostatics between these two atoms. Then when this particular atom, I mean, goes outside the cell, the corresponding calculation of the electrostatics will happen with the corresponding mirror image of the cell, and therefore this mirror image is actually helping the minimum image convention in the calculation of electrostatics. Same is actually graphically represented here for maybe a transmembrane kind of a protein. So I pictorially represent you the APP I had shown previously, the APP complex. If you undertake I mean protein membrane simulations and generate structures from Charn-GUI, you can see that even though you solve it, the water I mean the positive z-axis to a greater extent, in due course of time, as per the Charn-GUI algorithm, it will be equal number of water molecules in either I mean in either extremities of in the z-axis, in the positive and the negative z-axis along in the lipid membrane. So it may seem that this particular protein is then outside the box, but then that is not the case. When I have actually made the periodic replica along the z-axis, I can see, and the y-axis, I can show that it is within the the continuous explicit water solvent environment. Now, some initial analytical concepts I have actually I will discuss here. It will be essential when the hands-on session will be there. So we calculate, I mean, initial analysis like radius of gyration, which is the summation of the distance of each molecules with respect to the center of mass of this particular system of interest. Root mean square deviation is that is the summation of the square is the summation of the square of the distance between the particle at the time of the particles at each corresponding time, and same is the root mean square fluctuation, where the variation is actually calculated as a function of time and not as a function of residues. Now we will also discuss in detail, I mean in briefly, time correlation function. Correlation is like the product of two different parameters, and so if the correlation value is greater, is more than 0.8, that means suppose the RG, the radius of gyration, and RMSG, they can be correlated with each other, or they cannot be correlated with each other. So the Xi can be the RG and Yi can be the RMSG. Now the time correlation is when this the variation of this Xi is actually dependent on the time, and therefore the correlation of this Xi variable with increasing time, how it actually is correlated with the initial time of the parameter Yi. So this is the correlation function, but then there is an autocorrelation function which effectively is, it is the correlation of its own property. I mean Xi and Xi. I mean, suppose there is a particular property called rotational relaxation of water molecules. So rotational relaxation at time 0 and rotational relaxation at time t. How each of these are correlated with each other. This is very important in actually elucidating the dynamics of the hydrogen bonds or the surrounding water molecules in the hydrogen layer. So this is one of the examples. This is the rotational correlation function, and similar to the time correlation function, there is a pair correlation function, which is the correlation function of different moieties in space. So one of the examples is the radial distribution function. So details of it is given in this particular link, or there are other study materials which you can actually have in background while undertaking the, while you are getting expert in the simulation works in due course of time. Now I will come to the concept of drug discovery. drug discovery is a process. I mean, you people will be more aware regarding the drug discovery cycle and what are the nitty-gritties of drug discovery. I myself am quite new in this particular field, but even then I give some generic overview. Drug discovery is a process where candidate medications are discovered traditionally or serendipitously, and modern drug discovery is actually costly. It involves funding from different organizations, and it controls various mechanics, I mean, in the industry or academia, etc. And there are, different techniques of drug discovery. And so since a perfect drug candidate unlikely to obtain in early screening runs, in the high throughput screening actually is the process of finding new drug against chosen targets. It determines the sensitivity of the chosen target. But so other kinds of analysis like structure activity analysis relationship improves specific features, like it helps in determining the activity against increasing the activity against chosen target. And it reduces activity against unrelated targets, and it improves the ADME properties of molecules, the activation, digestion, metabolism, and excretion properties of molecules. We can get that from the structure activity relationship. And de novo drug addition is actually prediction of chemicals to fit in chosen target. And virtual screening and computer-aided drug design involving molecular dynamics and molecular modeling and molecular dynamic simulations have often been useful in improving the potency and properties of new drug molecules. So, I mean, since I have discussed a lot in amyloidogenic drug discovery and in the protein folding phenomenon, I actually want to focus on the relevance of MD simulations in amyloidogenic drug discovery. Why? Because the amyloidogenic systems by itself lack a three-dimensional structure. So the challenge in actually designing or discovering drugs in this particular domain is much more, and therefore because of these kind of systems, we need to understand in generically why the protein folding concept should be essential to understand initially if we are undertaking MD simulations and other molecular modeling kind of techniques in elucidating drug discovery related problems. So these are the general, analysis caused by we have discussed it, and still unclear. MD simulations can provide details of the misfolding and aggregation process because it can precise the pathway, the time, the time dependent pathway of the evolution of the ensemble with due course of time. And MD simulations can improve the enrichment factor of virtual screening by considering, because virtual screening happens, if you know, it will happen based on some crystal structures. If there is only one crystal structure, then based on that particular crystal structure, an MD simulation study can be undertaken where the conformation multiple conformations can be selected, and therefore the enrichment factor of virtual screening can be undertaken. By giving a more accurate ranking of heat compounds in combination with binding free energy calculations. And to improve the efficiency of lead optimizations, MD simulations can be used to obtain accurate binding modes and binding abilities of compounds and to uncover the mechanism of drug resistance. But limitations of MD simulations are existing force fields, especially in case of new molecules, new targets, and amyloidogenic proteins, which are actually devoid of secondary structure. If you go to the previous example, if you study this particular review article, you can understand, how the force field is relevant. The development of force field is relevant for addressing problems related to intrinsically disordered proteins. So whatever we are calculating, it is depending on already defined parameters, as I have defined, we have discussed in the force field section. So whatever the structural changes which can happen for amyloidogenic proteins or some other proteins, those can be, I mean, the force fields can have some secondary structural or some conformational bias. It can have helical bias, it can have beta sheet kind of bias. So the force fields are again, I mean, prone to correction and addition, modification. And so what happens is, because of the time limit, because of the computational explanation, because protein folding happens, or the drug discovery, when the protein and the drug is actually diffusing towards each other and fitting to the specific active site pocket, it may take quite some time, and therefore the ensemble to be generated by eventually will lead to loss, great amount of time, and that is one of the limitations of the normal MD simulations. But for systems where the definitive active site pockets are there, quantum mechanical tools like QMMM can be implemented for enzymatic active site study. Now, there are, in the drug discovery aspect, there are these initial techniques which are Rosetta and other techniques where the docking studies or some other kind of studies actually actually detect the drug. And as I have discussed the ensemble docking. This is the simulation structures based from one X-ray structure, or there are multiple crystal structures with which ensemble docking can be done. So the active site or the scoring factor will increase for the drugs with this. And receptor flexibility is this molecular dynamics or other simulation techniques. So some of these will actually I will discuss in terms of what is it called, of in decreasing the simulation time. One of it is the accelerated molecular dynamic simulations, where a boost, if this is the particular potential energy landscape of the particular protein ligand interface, then boost energy is applied which is dependent on the parameter alpha, which will actually decrease the barriers, the corresponding thermodynamic barriers, which I had actually discussed in the protein folding phenomenon, and therefore a protein may have ease in actually overcoming the barrier and going to some other conformational minima. Where maybe, so therefore different different conformations with respect to the drug, with respect to the protein ligand interaction is actually will actually be feasible in lesser number of time. Similar kind of a process is metadynamics. Like if this is the conformational space and the protein ligand is actually in this local minima where it may be separated with each other, or it may be at a conformation which is not the global minima. So it will there will be a force which will be actually applied to that particular system, so that it increases, it has its energy, it gains energy, and then it does, whatever conformations it has actually attained in this particular basin, it will not attain further in due course of time, and it will go to some other local minima. And how the energy is actually is given is actually based on some collective variables. It may depend on some specific secondary structural propensity, or the distance between the protein, the active site, the supposed active site, and the center of mass of the ligand, etc. So this is the metadynamics. This is the concept of the metadynamics. And then there are other expensive techniques which are the alchemical transformation, which means that means since the free energy is actually a state function, it does not depend on the path how it is attaining. This will also be a computationally expensive, that it has to be given energy and it comes to some other local minima where we may get the minimum, the more stable conformation in terms of protein ligand interaction. Here we already feed the ligand or the specific molecule of interest in the biomolecular active site pocket, and then we calculate the individual free energies of this particular, we calculate the binding free energy. We calculate the difference in free energy from when they are apart and when they are together, and this is the del G bind. And then we actually dissolve or evaporate the ligand. So it will be eventually, this is the the solvation free energy again in water, and this will be the the solvation free energy of the protein. From this particular free energy, if we dissolve the ligand, it will give the solvation free energy of the complex in solvation free energy of water, and this will give the solvation free energy of protein. And this particular phenomenon where the dummy ligand, which is not existing, is actually binding to the biomolecular surface, will actually give a del G is equal to 0. And combining all this, we can get the del G bind to be the del G water minus del G protein. You can get a more detailed explanation in this particular reference. And also, in terms of alchemical transformation, there are other techniques, like if we want to change, see the scoring functions of different drug moieties, we can change the we can tune the thermodynamic properties based on, by altering the suppose a particular atom, suppose from nitrogen to carbon, or some or methyl to some phenyl group, etc. And likewise, we can also study the del G, the free energy of binding, and thereby the scoring functions of the respective molecules of interest of maybe of specific specific classified molecules. So later on, there is another technique between the docking and scoring functions and these expensive molecular dynamics techniques, which is the molecular mechanics PBSA, for Poisson-Boltzmann surface area, or generalized bond surface area. These are, essentially based on similar kind of mechanism, but the modeling of the solvent is dependent on two different equations. Two different approaches. One is the Poisson-Boltzmann approach, and is the generalized bond approach. So essentially, the ligand plus receptor will actually join as ligand receptor or the ligand protein complex. So the del G binding will be the difference of the free energy of protein ligand minus free energy of protein minus free energy of ligand. This G will be actually the bonded and the the bonded and the non-bonded terms as dependent on the force field, as well as I have discussed that the free effective energy depends on the enthalpical and the solvation free energy terms. These are the polar and the non-polar components of the solvation free energy of the system, and this is the entropic term. So overall, this is the effective energy, and the addition of entropic term will give you the effective free energy. So this del G bind, so this G is actually defined by this particular parameters. The del G bind, therefore, will be the G, the free energy of protein ligand when we are simulating the protein ligand separately, and minus free energy of protein when we simulate the protein, and minus free energy of ligand we are simulating the ligand. All these are separate simulation systems. So this is a three-dimensional MMPBSA or MMGBSA. But for ease of calculation, we can do that del G bind is equal to GPL minus GP minus GL from the same protein ligand simulation. We only calculate, suppose we can calculate the free energy of protein ligand together minus the free energy of protein in due course from the conformational ensemble only by selecting the proteins, and similarly only by selecting the ligands. Now, with a particular biotin variant, BTN1 to BTN77 variant, we find the different corresponding the binding free energy terms and the effective binding free energy for the different variants. But we see that the standard deviation and the standard error are quite high. That means the MMPBSA and MMGBSA can be inaccurate. But even, I say here that the MMPBSA and MMGBSA is actually more frequently used in terms of implementing MD simulation studies in drug discovery or this kind of aspects. So, so it is found that this error can be minimized if we take multiple, I mean, instead of simulating for 1 microsecond or 1 millisecond, we can simulate 10 separate simulations of 10 microsecond each, and therefore something like that, and therefore it will maintain the conformational space, conformational ensemble, equivalent conformational ensemble, but it will decrease the error in computing the binding free energy. And then again, this is a one of the studies in my previous group where protein-protein interaction is actually computed with again the effective energy and solvation free energy. You can actually go through this particular paper and some other relevant papers in protein ligand complex of Professor C.N. Ham, and you can see, and this is another another way in computing the free energy of protein. I mean, the solvation free energy will actually denote how effective the binding of a particular ligand or a particular protein or a particular substrate is in another protein or another biomolecular substrate. You can go through some of the papers of Professor C.N. Ham. And since due to the lack of time, I will stop here. There had been a few case studies related to protein folding problem, but I will stop here. Thank you. And if there are any questions which I'll be able to answer, I'll be happily attending that. Thank you.
Questions from the participants are now welcome. Please unmute yourselves and ask a question, please, if you have any.
Hello, sir.
Yes.
Thank you for the very nice presentation, sir, and very good morning to you, sir. My question is regarding the solvation energy. Like,
Okay.
as from the biochemistry, we know for protein folding and its activity, the solvation is important.
Yes.
And
Yes.
that solvation will depend on the what kind of solvent we are using and what kind of solvent the protein is stable in. Like some protein are stable in organic solvents or inorganic solvents, and some protein are stable and active in aqueous solvent.
Yes.
But as from the presentation, I see that only all the force field they include water molecule.
Yes.
Like like they provide the charges for water molecule only, and they assume that it is soluble in water. But what if we want to solvate our protein in some other organic solvent or inorganic solvent? How can we proceed in that case, the molecular dynamics?
Yeah, actually, for most of the protein folding cases, water is taken to be the ubiquitous solvent because much of the biological reactions happen in water. So, I mean, first and foremost, I mean, for protein folding phenomenon, generally non-polar solvents or semi-polar solvents are actually taken if we want to maybe generate some specific structure of protein which is not, I mean, suppose for amyloid beta peptide, which I have discussed. For amyloid beta peptide, it is intrinsically unstructured, but it happens to get helically structured in presence of lipid mimicking environment. And for those cases, I mean, the protein conformation is actually studied in the, experimentally and computationally in different non-polar or semi-polar solvents like HF5P. The reported PDB structure of amyloid beta 42 is maybe in HF5P, hexafluoroisopropanol. So, this is, I mean, this is the what is it called? I mean, this is the relevance of the non-polar, semi-polar solvents in case of protein folding. In coming to your question, how, I mean, we have to check the parameters which have been defined in the force field, whether it is already, already capable of calculating the parameters of some organic solvent or not. But generally for protein folding process, water is coming into play. A priori, I cannot say, because most of the cases I have found that it is coming from the studies happening in water mediated interactions and protein folding, protein. If I could have shown some of the case studies, we could have seen that how the internal energy and the balancing the internal energy and the balancing the solvation energy is happening with an overall effective decrease in the effective energy. But since due to the lack of time, I am actually omitting that. Yes, and there has been another question: what is the difference between MMGBSA and MMPBSA? So it depends on the two different equations. One is the Poisson-Boltzmann equation, and another is the generalized Born equation. I think in the generalized Born equation, the dielectric continuum of the solvent is actually defined, already predefined, which is around 80. I mean, in spite of 78.2, I think you may check. I may have to educate myself more. Generally, people tend to give it to be 80 for water, especially, and so they provide this dielectric continuum and a particular what is it called? Particular ionic strength, 0.15 ionic strength, etc. And Poisson-Boltzmann equation depends on some, maybe depends on some other factors. I have to check that. I cannot say that right now.
Okay, I just want to add a few things for the first question. Actually, it is not right that all the force fields are for the water molecule, but if you know that your protein is solvated with some organic or inorganic or some ionic liquid, then maybe you can design your force field like that. You have to design that parameter for particular type of organic molecules. But you must know what exactly you are going to do. Like you can check my one publication with where I designed the new force field parameter for an ionic liquid, because that particular protein was happy with the ionic liquid. And also in another paper, we have also worked with like lipid water molecules. So there are several things you can design your experiment if you know that what is the preferable solvation for your protein, so you can design. So there is no foundation. Thank you.
Yes, but actually for the solvation free energy, maybe there might be, since it is happening for already defined force fields, so that needs to be taken into account. Maybe solvation free energy, which for normal protein solvent studies, it is absolutely okay. But generally, solvation free energy calculations happens with resin calculations for AMBAR, or maybe I do not know, MBAR with Gromos implemented force fields, GROMACS. So for that, this need to be checked, maybe.
Hello, sir.
Hello, sir.
Yeah, there is one possible one. Is there any possibility? Sir went off. Pradeep, is he there?
Hello, sir.
Sorry. Yes.
One question, one simple question.
Yes.
Yeah, sir, the post-translational modifications of proteins like ubiquitination and metallization and all those things, will it happen during entropy or enthalpy-driven stages, sir?
I think it will happen. Evidently all are actually happening with a pre-diffusive mechanism. It has to approach near towards each other, and that is actually gone by the long-range electrostatic calculations, which we try to implement in our calculations, and therefore the MD simulation studies. But eventually, the more of the stabilization will happen in terms of the enthalpy-driven process. I suppose, I presume.
Thank you.
Sir, Pratit,
Yes,
sir?
yes.
We have a question in the chat box from Bharat.
Okay.
How cofactor MD simulation parameters included in simulation?
Cofactor simulation parameters. Actually, cofactor. So it depends on. For generic force fields, cofactor may be some metalloids or some other things or some other moieties. So for if it is metal ions like calcium, zinc, etc., those are already defined in the force field. But if it is some other small molecule, then we can design it by collaborating with some experimental group and then quantum mechanically calculating with DFT studies. Or there are, for CHARMM, there is a database called SwissParm, where you can actually give the particular structure of the structures of the, what is it called, of the molecule, and we can get the particular parameters of the force field. And what all parameters we need to know to define our solvent for solvation. So what all parameters we need to know, meaning, I mean, same parameters I like the bond angle and the partial charges, bond angle and partial charges for a 3-site water model. And for a 3-site water model like TIP3, the melting temperature is quite low, maybe 172 Kelvin, which is unnatural. So if you undertake cold denaturation studies, MD studies, you can implement TIP3 water model. But if you tend to have more realistic, tend to produce more realistic results depending on force field. The cold okay, yes, I will share the presentation. So if you have if you want to reproduce data more relatable to experiments, you can have more other water models like TIP4P, where a non-bonded site is also present with a dummy chart, something like that, and then TIP5P as well. But generally, till that, I mean, the most developed, each water model has its own consequences. Like for studying the antifreeze protein, we can maybe use TIP5P, but TIP4P evolved and TIP4P 2005, these are now more generically used for maybe if the system size is not, more bigger, we can use those water models to, more accurately reproduce the data. Now, how do we include the effect of pH in MD solution? Adding hydrogen ions. Of whatever, if 1 mole of hydrogen ion, what will be the number? What will be the particular number of hydrogen ions? In a particular box, and then correspondingly adding it. We can add randomly, or we can add with some particular techniques.
Thank you, sir.
Yes,
And now,
thank you.
I now request Dr. Kripanidhi to give some concluding remarks and his gratitude as well.
Thank you. Thank you.
What is Sanjana?
Sir, your concluding remarks.
Okay. Hello. So thank you all participants and experts. Good morning to you all. And we are immensely happy to have Prateek Chakravarty, and he has given a wonderful talk on protein folding. Protein folding is quite interesting because it is the one that is involving in much of the metabolisms in our body. So he has explained very neatly about the entropy, enthalpy, and another interesting thing is Lewinthal's paradox, wherein for one amino acid in the peptide chain requires 198 conformations, so that it will take enormous time. So that's what now, there is no synthetic protein so far. They're all having biologically driven proteins that have been synthesized now. Okay, so now he also made a good presentation, and with regard to the drug discovery, and still a lot to go with regard to this protein folding and enzyme mechanism, wherein we wish to synthesize an enzyme that targets the pollutants. We keep them in our environment and atmosphere. So for that stage, we have to reach. Thank you, one and all. Dr. presentation.
Thank you, sir. Thank you, Dr. Chatterjee. We will now have a hands-on training session on MD stimulation of apoprotein with Mr. Krishnendu Bera, who is working as a research specialist in the Czech Republic, and also Dr. Chatterjee as well.
Okay, thank you, Sanjana, for that nice introduction. So shall we start now, or we have 10 minutes break? Then we'll start, because the original time was from 10:30. So
yes, we will go for a 10 minutes break, Krishna, as you suggested.
Because this
As the live is no problem. As the live is running, it's still we kept same. No
Yeah,
problem.
yeah.
Participants can have a tea break.
Yeah, sure.
Yeah, they can again join by 10:30.
Yeah.
Meanwhile, those who did not have installed all the software, required software, I'm strongly suggesting to install them. I think many of the participants are aware of using the Linux.
Okay.
And also we have sent the instructions regarding to install the Linux app in Windows 10 as well as VirtualBox installation in Windows 7 and 8 systems.
Mhm.
A lot of videos are available. No, it's not an issue. And also participants have they got a good time to practice by seeing the recording sessions. I think it's not an issue.
Okay, maybe if in the meantime I can ask that question, that how many of the participants know is there anyone who doesn't know about this Linux command? They can raise hand. We can have
Go ahead.
Then maybe I can cover that part as well. Because I have a belief that most of the participants are from the biology background. They don't have the computational background. So maybe.
Yes. Compact. Can you rename yourself? What is your name? Compact.
So myself, Shefin. I'm new to Linux operating system.
You are using Windows operating system.
Yeah, I use Windows operating system. I'm new to Linux operating
Yes.
system.
Did you get the instructions? Did you get the 2 files which is containing
I have
that?
got the instructions. That I have downloaded.
One by one. Ajit Singh, please mute one by one.
And it will be more good if you could tell the basics too.
Yeah, Krishnendu.
Okay. Then maybe I will cover the little bit of Linux command, so that it will be useful for most of the participants. So that's
What he's saying is he's having Windows 10 system. Is it right?
Yes,
Yes. Okay. Did you manage to install the app, the Linux app?
yeah, I'm doing
Yeah.
it.
Yeah. Can you please share your screen? What's your name? Can you rename it?
So myself, Shefin.
Shihrin.
Yes.
Okay. Can you share the screen?
1 minute, sir.
Yeah. In the meantime, other participants who have got the Linux installed can take a 5-minute break.
Sir, this is what I know.
Yes. That's fine. Krishnan, can you able to see? Yeah.
Yeah, I can see.
You give any name, Unix username, and followed by password. You remember the password. Sheffield, good. Password won't be visible. Yeah, enter. Just enter. You give the password, normal any password 1 2 3 4 5 6 like that, but remember that password. It won't be visible, but you remember. Yes, very great ability.
Yes. Just Y, enter.
Yes, good. Just you update it, sudo apt update.
wait for 1 or 2 minutes because
Yeah,
it is installing some repositories.
Yes, Rohit. Yes, Rohit. Thank you. Thank you for assistance.
Now update the system.
Just type update.
Yes.
No,
sudo.
no.
Sudo.
Yes.
App. Give the password. Hello, Dr. Ajit Singh. Do you have some problem, Ajit, on installing this? Do you get your system installed? The Linux operating system. Ajit Singh.
Hello, sir.
Yeah, tell me, Jyotirmay Dev. You can tell.
Sir, actually, I am facing to install GCC.
Okay, you did you give the command sudo apt get install GCC?
Yes, sir.
Yeah. What did it say?
Sir, can I share screen?
Yes. Meanwhile, Shefin, you go ahead with the commands. You're typing the commands.
Okay. sir.
If any error will come, you just again unmute yourself.
Stop
Okay.
sharing.
Can you stop sharing? Yes.
Thank you.
Yes, please share, Jyotirmay.
Sir, please finish.
Yes. Will be visible. Just started sharing. It will be visible. Yeah, now we can see. Okay. Go up, go. Oh, okay. Again, you type the command sudo apt update. Sudo apt update. Not get. Okay. Type. Get. No problem. Type get. Update. Okay, give the password.
Yes, sir.
Oh, again it is coming like that. I think some error with that cleaning part. You try app get clean. Rohit, can you comment on this?
Yeah, actually, sir, it is not getting the mirror from where it is trying to copy the update files.
Yes.
So it is not getting the mirror. Maybe he has some internet issue. How you are accessing the internet?
What, sir?
How you are accessing the internet from your Wi-Fi or from your?
From a mobile Wi-Fi.
Okay, so you should not need any proxy or some other things. Okay, just reboot your system and then try to update the Ubuntu.
Okay, sir.
Okay. Then
Okay.
because it is not getting the mirror from where it is trying to get the repositories of update. Okay.
Okay, sir. Thank you, sir.
And if you cannot update, so you can do the basic Linux command, but you cannot install the Chromax without updating. So try to update if the problem is there. So we will tell you some.
Okay, sir. Okay, sir. Thank you, sir.
Yes, Anil Panwar has given some suggestion. This problem of normal counts when user use proxy internet. Yes, might be the reason. I think Krishna.
Yes.
Yes, it's 10:30. I think can we start?
Yes, sure, definitely.
We have so many more sessions today. Night session also there. We can.
Yeah, actually the thing is, before I start, I just have an advice to all the participants that please do not run the long simulation in your laptop, because if you love your laptop, then please don't. I tried once and I have to dump the laptop. It was only 10 nanosecond simulation and it was just, you can say that it burned. So please do not try. MD simulation is really very robust and very, the computational costing tool, so that if you will run in your system, in your laptop or in your personal desktop where the hardware is not that much good, so please do not try there. You should have some server, at least some workstation, like where the hardware is better. So try there with proper cooling, but you should not try your like local desktop or laptop. So
Afrin Khan is asking about installation instructions on GPU. Yes, you have. I think you need to have CUDA libraries installed.
Yeah, that is not necessary to have GPU. Like if you want to accelerate your simulation, like let's say you want to get some speed of the simulation, so that time you can have GPU. But it is good to have GPU because all of the these days these softwares are enabled with the GPU, so you can just speed up with that. So please use some good hardware, and if you have some supercomputer or some servers, so please use that. Okay, so you all have like very like theory session in the morning, so maybe you will have some idea that okay, simulation is just about some equation and nothing there. But simulation, I must say that simulation is really beautiful. You can solve ton of problem which you can't solve by the experiment, so you can solve here. And like you can have many problem, like biological as well as chemical problem. In all the field you can have some like really very interesting results. And I must tell you that you should have your some I mean, use your brain to interpret the data, because anything you can simulate, but if there is not biological relevance, so that is completely nonsense. So please, I mean, get the results, but use your own brain to interpret them and correlate with experiment always, because the experiment is the ultimate, I will say that validation. So here, every time you should correlate your data with the experimental data. So now I can share my screen. Okay, just give me a moment. So can you see my screen?
Yes, Krishna.
Okay, thank you. So I don't need any introduction, so I will go with, so I just prepared some Linux basic commands. So those who are not familiar, I mean, this will be useful, and those who are familiar with them, so please bear with me. And before I jump into the Linux command, I just want to show you one video. Exactly, this videos are created by, I mean, just next to my lab. They are really doing wonderful work with the simulation. So please have a look this video. Okay, so here let's assume that these are the two confirmation, and
You're playing any video, Krishna?
Yeah, I will.
Oh, sure.
So these are the two confirmations. Just imagine that you have your protein. Just imagine that you have your model. That this model is like this is the final confirmation. Okay. So now in the simulation, what will happen? You will just put into some protein into some the virtual box, and give the local environment. There will be some environment like in this, just imagine the protein inside the cell. They have like water, they have some temperature, they have everything. So now at the end you will get your I mean the final confirmation. So this is the way we actually do. We take some random confirmation of protein, and we want to simulate them, and at the end we get some confirmation, and we correlate this data with experiment, and this is like here we get the confirmation. So this is like a grandma way of teaching simulation. So, and here I want to show you that there is one problem, like this membrane protein, they have like they form the pore to facilitate the different kind of I mean substance to inside or outside the box. So they have like really very simulate this. This is the lipid bilayer, and this is the protein.
Krishna, are you showing any video? You couldn't able to see. We just see.
No. I'm just explaining.
Oh, okay. You will be showing video afterwards.
Yeah.
Fine. Okay.
So this is the I mean this is the bilayer of the cell, and this is the protein, and you can see that during the simulation how it will make the pores. So this is really wonderful, and I mean this is one example of doing simulation. You can understand the I mean the beauty of the simulation. I mean you can solve like this kind of problem with help of simulation. So I just want to have some feeling related to the theory that, okay, we are not doing anything, I mean, like some like equation or some theory kind of stuff. We are also visualizing this protein, and we are solving the real issue. Okay, so now I just want to start with the commands. So, now I am in India, and due to some family issue, I'm stuck here and I can't return back to Europe because now the COVID thing and the travel restrictions, so I can't go to my lab. So the problem is how I will work. Like they are giving me salary, and then the end they need some work done. So now I'm here. So just assume that you are in the I mean, you don't have the access of lab, you can't go. This is really a corona situation. And now we are like, this workshop should have like in physical workshop, but now it is virtual. So how you will work? So I will show you like from India, how I will connect to my computer, and I will do all the work there. Okay, so let's start. So this is my computer here in India, and I will connect to Europe to my supercomputer. Okay, so for that you will need some specialized commands. So how you will connect your local machine to your server. So this is SSH command, which for which we can connect to the remote computer, but it should have the access. I mean, this.
Krishna, you didn't have share your full screen. We're still seeing the Unix and Linux command.
Yeah, I'm sharing only the Unix and Linux command.
Okay.
Yeah, I will show you.
Okay.
So how I mean I will connect to the local machine. I mean this remote machine. Okay. So can you see my terminal right now? So this is the terminal. Can you see?
No.
I guess you have not shared the entire screen.
Yes, Karan, I
said. Yeah, maybe you have to share your whole.
You have shared only PPT.
No, wait, Karan, wait.
Okay. Maybe I will share again. So now can you see?
Yes, Krishna.
Okay, so this is my local machine where I am right now. So I will connect to my supercomputer. So for this you need SSH command, and if you want to visualize, the GUI version, like you want to visualize something from the remote machine, so you will use Y, capital Y with flag. And also if you want to execute something from there, so you will use X. So basically I recommend to use X and Y both. Then your username, like this is my username, and at the domain whichever provided to by your admin. Then give the password. So now I am connected to my machine. I mean this remote machine. Okay, so this is my supercomputer where I basically do work. So from there, I actually connect to my lab machine. So this is Ricky is my computer. So I will go to there and I will show you. So basically, I have created one directory for the today lesson. So okay, so I'm here and let's see. Okay, so the next command is SFTP. So now I have connected to the local machine. So if you want to download something, let's say you want to download some file, let's say here, I will create some file. So this is some file I have created, and you want to download it from the local, to remote machine to the local machine. So for that you will use SFTP. So I basically recommend SFTP because this is with the FTP protocol, so the download speed will be very high. Because you can also use SCP. There is also another command called SCP to copy the command, but when you are downloading some bigger file, let's say your file is, let's say, with some 10 GB or 100 GB. So at that time, the FTP is really very faster because it downloaded in FTP protocol. So use the SFTP, then again the same command, like your user ID at the rate the local, the domain name. So my domain name is Onyx, and let's say I will download this A file to my local machine. So okay, so I will give the password. So now you are connected. So for uploading, you will use put A. So I want to transfer A to the local machine. So you will use put, then use the file name. Okay. So now it uploaded. So let's say you want to download something from the local machine. So you will use get, and let's say I will download some file already exist in the local machine. So let's say pdb is there. So I want to download it. So it will download to the remote machine from the local. So
Krishnan, one question from our Kalyani madam.
Yeah.
Why are not using rsync for bigger files?
Yeah, you can use the rsync, but the thing is here with the STP, actually the beauty is you can use all the command also. Let's say you want to change the directory, so I can use any command here as well. Like I want to see the it will not work. So change the directory. Let's say I want to go somewhere else, to the home or somewhere. So you can use the SFTP as for the command line purpose, because for the rsync or SCP, the problem is you have to know the prior knowledge of the path. Basically, that is the problem. Every time you have to open the two terminal and you will know the path. So, but here you don't have to know the path. You can use the any basic command and you can use it. So basically this is more, the more friendly. Sorry, something happened. It's hanged. Okay, now it's working. I'll use another terminal. Okay, so you can see the solve is here. So basically, you download the this solve PDB from the local machine to your remote machine. So am I clear? This is like you can ask questions, so you can stop me anytime. So is that clear to everyone?
Yes, sir.
Okay. So the next is ls. You want to know what are the content in your directory. The folder. Basically, you will use ls. So it will list all the files, directory, whatever in your machine. So you can see that it listed. Actually, if you want to just a minute, it's not working. Okay, I'll show you. Okay, so if you use ls, so it will just list like randomly but I really like to use ls minus ltrh. So it will list all the file as well as that the user ID as well as the permission of the file, and also the file. I mean this how to say? I mean the memory of the file. Let's say.
Krishna, Anil, Krishna, please. Anil is asking, please tell again how to log out remote PC and connect to your local machine terminal.
Okay, so just use exit, exit. Actually, this one is like freezed, so I could not show you. Okay, so I will show you. So you are here, so use just exit command. That's it. So now let's say you are in the Riki and you want to log out from the remote machine. So use also exit. So now, okay, so something, yeah. So now you are in the script. So previously you were in the Riki. So now in the script. So if you want to come to, this is also another supercomputer where I log in. So this is the interface basically to log into my institute. So if you want to come to, my computer in India, so again exit. So I'm here. So is that clear?
Yes, you continue.
Okay.
Okay, Krishna, I want to mention something.
Yep. So.
Okay, the participants who didn't use Linux, actually, they need not worry about that. You have to search many things on the internet if you are first time in the Linux, because Krishna, if you are seeing the first time that Linux terminal, so you will get confused. But if you want to do the bioinformatics, so you have to work on Linux, okay? And if you are using Windows also, so like here he's shown SSH command, you can use putty also to access the server. And for the SFTP command or any file transfer protocol, because our ultimate aim to copy the file from server to our client computer, so you can use WinSCP also, okay? And one more thing I want to tell you here, that if you are using your local machine, so no need to connect the server. If you don't want to connect the server, so you don't need SSH and SFTP command, okay? Rest of the command which will stored by Krishna, you need those command to perform the operation. Okay, continue, Krishna.
Okay,
thank you, Rohit.
Thank you, Rohit. Actually, if you are in the Windows machine and you want to have the interface of Linux, so you can use this mobile external software. So this will give you a Linux terminal where you can use, like you can give all the this Linux commands, and you will get the interface of Linux. And for that, you don't have to use the putty, and this putty is just for login, and WinSCP just for the copy of the file. So you have to install two software. So, but with the mobile external, you can do everything in one software. So you can use. It's really very friendly. Okay, so we'll continue with the command. So now if you want to change the directory, let's say I'm in the workshop directory and I want to go to the directory of FOMD. So you have to use change directory, that is CD and FOMD, and enter. Now you are in the FOMD directory. So how you will know the path? That is another command that is called pwd. So here you will know the path of your, the current path of your directory. So then the touch command. Touch command is if you want to create some files, so you can use the this touch command. Already I have used this. like to create the A file. Let's say I will create another file that is B, and you will in that way you can create the file. So here you can see the B file has been created. So now if you want to delete something from, delete the file from your directory, so you will use rm command and the file name. So you can see the file has been deleted, B file has been deleted. So I will recommend you please use rm command very carefully, because in the window we basically use click, do the right click to delete the file or something. So here if you if you lost the file, so you can't retrieve, it is really very painful to retrieve. So please use this rm command very carefully, because I have seen that some of my colleagues just deleted entire PhD project like in rm. So please be careful. So if you can also ask your administrator to give some restriction of using RF, like in our institute, our admin has given restriction, like I will show you how. You can tell your the administration to give the restriction, like, okay, so there is a file and I want to use I want to delete it. So it will ask you, this is a regular file, you want to delete or not. So this is the restriction which I was talking about. So this admin can give the restriction. So if the user will give yes, then it will be deleted. So here you can see this is deleted. So this is also good practice to ask your computer administration to give the restriction when you are using the Linux interface. Then you want to create some folder. Let's say, first you have to know that what is file and folder. File is just something where you will write something or some text file or something like that. The folder in the Linux we call it directory. Directory is just same as the folder you have like in the Windows machine. So in the directory you will keep some files basically. So you should distinguish what is directory and what is the file. So if you want to create some file, so that is mkdir, sorry, the directory. Let's say a directory you want to create. mkdir, so directory name, and enter. Okay, so it's already a file is there. Let's say AA, and it will create the directory. So basically, how you will distinguish between the directory and the file. Basically, the thing is, in some of the Linux, like Ubuntu, they have like different color code, but I am using the CentOS, so there is no such color code. So you can't, some visualization you can't visualize. So basically, you can use what I use, like I use a cd command to to know it. Like you can enter to the directory, but you can't enter to the the file basically. So then, how you will remove delete the unwanted directory. So if there there is two things. If your directory doesn't contain anything, so there is rmdir command. If your directory contains something, so you have to use another command. I will tell you later. So let's say your directory doesn't contain, this AA directory doesn't contain anything right now. So you will use rmdir command, then the name of the directory, enter. So it will delete the directory.
Yes. So now we have a question, Krishna. Kalyani ma'am, just unmute yourself.
Thank you, Abraham, and thank you, Krishna. we can actually create a color code for any terminal, even if it is on cluster or CentOS. It's a one-line command code that I can happily share in the chat, and there is a possibility. So it's not completely impossible, but we definitely can use color code for terminals even on HPC. I'll share the link for the one-liner. Thank you.
Thank you, Kalyani. Kalyani Dusya madam is also one of our invited speakers. She is going to deliver her lecture.
Thank you, Kalyani.
Thank you.
Okay, so let's say if this ApoMD, I will create another directory. Let's say AA, which I have created, and I will enter, and I will, let's say, keep some file here. So here the directory containing some files, so you can delete the empty directory with RMDIR. So it will give you error message that the directory is not empty. So for that you will use rmrf command. So if your directory is, it contains some files, so you will use this one. So enter. So now your directory is deleted. So basically now the thing is how you will copy some file to one directory to another directory. So assume that you want to copy the a file to some directory which already I have created, like this is ApoMD directory. So if you want to copy that file to some directory, so you will use like this, then enter. So you can see the A file is here, and I will enter to the FO file. So you can see that FO file also having this F file. Okay, so now I can delete it. Okay, so if you want to go to the previous directory, so you will use CD /. So you will go to the previous directory, but if you use CD, it will take you to the home. So now you have like a file here, and you have deleted the A file in the ApoMD. So now if you want to copy one directory to somewhere else, so for that you can't use just only CP command to copy your directory. So for that you have to use CP, and another is CP and the R flag. So I will create one directory, let's say AA, and I want to copy this directory to ApoMD. So now it's been copied. You can see this AA directory is here. So like I can delete with RMDIR because this is empty directory. Okay, so now if you want to move some file or rename some file, so let's say you don't like this file name AA already, you can see here. So you can rename it like into B. So this is the MB command. See, your file name is renamed to B. And if you want to move in the same command, you can move your file to some directory. Let's say I want to move this B file to AA directory. So here this B file is not here. So you will enter to the AA folder, and here you can see the B file is here. And if you want to know who are the which are the program is running in your computer. I will, for that, I will use the top command. So it will give you the people, those who are running like now. I'm using this, I'm the user, so the Firefox is running, then I have used this top command, it is running. So these are the thing you can get to know, like if you run some script or something, and you want to know the I mean what are the, I mean the how many CPU it is using or something else you want to know. So with the top command you can know it. And also the process ID, this is called the ID. And if you want to kill, I mean stop the code, so you will use this process ID, like you can copy or remember this ID, and with the kill command, like I will just stop it, and let's say I will kill like this Firefox. So I will just copy or paste, and I can just enter, then it will be stopped. I don't want to stop it, so I'm not executing. Okay, so till now everything is clear. Okay, so I can move to the next slide. So if you want to know the date, you can use date command to know the date and the time. And if you want to know the calendar, it can give you the calendar with cal command. And if you want to know, I mean who are the user ID. So here you can know with the user ID, like who am I. And if you want to know the size of the of your folder, so you can use the div flag sh command. So it will give you the size of the folder. So the size of the folder is this much MB. And if you want to zip something, let's say I want to zip everything in the directory, so I will use zip. And for the everything, you will use star command. And if you don't want, I mean just particular some file, let's say I will use solve pdb and AA. Okay, so it will be like one. Okay, just wait. Okay, so let's create some other file. Let's say A, and you want to zip it. Okay, so now you have zipped it. Okay, so now you have zipped this to file. And if you want, like if your computer is freeze and if you want to stop it, so just use Control plus Z, so it will stop everything. And as one user, I mean the participant asked us that how to log out from the system, so you will use the exit command, as I shown before. So I think this much is enough to, run the simulation. So maybe we can start with our real simulation. Okay, so before that, I want to say that there are many softwares available. Some are like paid software and some are like open source. So for this workshop purpose, we will use GROMACS. So GROMACS is an open source software, and it is like, people say that this is really very fast because this is, the history of the GROMACS is it developed in Europe, in Groningen University, with like the real developers are like my supervisor, they are the developer, they first developed GROMOS software. So from there, like the Gromos force field and everything they have developed. So then after a few years, I mean, they are separated, and some people, I mean, they develop Gromax, and the Gromos software is already exists, and the Gromax is with another group. So now we will use the Gromax software because I mean, it is faster than the Gromos, the older one, and it can compatible with most of the force field. I mean, I will say that all the force field you can compatible. So when you are, I mean, testing any force field, like which force field is really good and which is bad for your system. So for the comparison purpose, Gromax is really very, I mean, the great software, because it can give you you can use all the force field parameters, and you can compare your study with different force field. And also this, as I told you, this is a open access software, so you can have you can modify anything. Like in the morning session, someone asked that how you will design our own parameters. So I will tell you that you can design the parameters. So you have to know the force field first, I mean the equation of the force field. So from there you can design your own parameters, like the bonded and non-bonded things, whatever, I mean, we have discussed in the morning session. So this value you have to know. So you can ask that where I will know. So you have like two methods, like from the experimental value, you can know this bonded, non-bonded parameters, or you can do some quantum calculation, and from there also you can know, but this is not that much accurate. But I will suggest that please try to find the experimental value. From there you can add the parameters, and you can design your own force field parameters for anything. So now we will start our session. So before that, I just want to show you another thing, that is the time scale. Like yesterday, one of our participants, they asked like how much simulation we should run. So for that, I found this is really very useful picture from BioStar lab I found. Actually, from here we can see that if you're working with the proteins, so we are in now with the present computational, I mean this facility, we can run up to microsecond. Like nanoseconds are like 10 years before, nanosecond was like nightmare to run it. So now people are running in microsecond, and some lab like in US Pandey lab, they are running in the millisecond level. So you can see in the femtosecond and picosecond, we can see the vibration motion of any molecule. And from the up to nanosecond scale, we can check the rotational motion. And if you are interested in the protein, like you can see the side chain folding and some helical folding up to the microsecond, and also some loop dynamics up to the microsecond. And as this workshop is for the ligand, for the drug designing, so you can see the movement of the, I mean, the interaction of drug in the nanosecond to, I mean, if you want to see the realistic thing, so you have to go to the second level. And if you are interested in the protein folding, so most of the protein folding, if you'll see in the second or millisecond level, you can see the some domain movement and some real folding you can see. But this is not the case of the intrinsic disorder protein, as Dr. Pratit show us in the morning, that this intrinsic disorder proteins are really different. These are like unstructured proteins. So I have experience with one IDP protein that after 2 microseconds also, this is like nothing, like we didn't get anything. So for that, you have to bias your system, like you have to, like these are called like enhanced sampling. Like you will bias your system. But for the classical simulation, we are not biasing anything. But if you know the parameters, like if you know this, some parameters which are affecting your system from the, let's say you want to bias the system. So people are using metadynamics and many things for the biasing the system. For the main problem is how to know which parameter we should take for the biasing the system. Let's say people are using temperature. So temperature is a biasing, because we know that in temperature the protein will it will denature or something, like it will like it works. So people are using temperature as a biasing, or you have to know your the parameters. So there is several way to know the parameters. I don't want to go to that far. So there is also the enhanced sampling, which Dr. Pratit had shown us, like metadynamics. So for the metadynamics, you have to know this parameter, as I told before. So there are several parameters like RMSD or RG or other things are there. So you have to know this parameter, and you have to bias your system to reach the global minima. So as Dr. Pratit so asked, like the protein or anything really want to, in the realistic environment, they want to be in the low, lowest energy conformation for the stable conformation. So to achieve that, we are doing lot of things, like we are doing the classical MD, but the classical MD, the problem is the time scale, like from which time scale we will reach this global minima. So this is, really very impossible to say, but with the enhanced sampling, we have something like we can bias the system and we may reach the global minima. Okay, so I think you have some idea about the simulation and how we are doing. So basically, I can say that the simulation is just, I will give you another example. Like, let's say some billion-dollar car is developed by BMW company or some other company. They are designing some car. So this car is really very expensive, and they want to know that whether in the real accident case, the driver will survive or not. For that, they can't just do the accident and they will see that whether the driver is dying or is surviving. So they do the simulation. They create the virtual environment of the giving all the parameters of the accident, and they see that whether the car driver will survive or not. Like that, here we are just mimicking the cellular condition. Let's say your protein is soluble protein and cytosolic protein, and you want to create the environment of the soluble protein, like you want to create the virtualize the environment. Like, let's say in the cell there is some water, there is some ions is there, and there is some temperature, there is some pressure, lot of things are there. So you are just mimicking the environment and you are creating one virtual box, and in the box you are putting your protein and giving the environment and doing the simulation, and you want to know that what is happening to the protein in some certain time scale. So that is called the simulation. Nothing like some rocket science is here. So this is really very simple. So like one participant asked us that there is some proteins, these proteins are solvated in different mediums. So you must have to know your protein and in the environment where you want to do the simulation. So you can create that environment and you will put your protein into that environment and you will do the simulation. So that is the case. So first you must know what is the problem you are going to tackle. Then you will create that such environment for this protein, and you will do the simulation and get the result. I must tell you that whenever you are getting the result of the simulation, please I mean, correlate your result with the experimental value. Like there are a lot of experiment, like NMR is really very good when you are working with the intrinsic disorder protein. So there are certain parameters like NOE or let's say J couplings you can get from the NMR. So also from the CD also you can get some secondary structure information of the protein. So you can correlate your simulation data with this kind of experimental data. So you can predict the CD spectra from your simulation, and you can correlate that spectra with the CD experiment. You can predict the J coupling, you can predict the chemical shift, whatever parameters from your simulation, and you can correlate that result with the experiment so that you can validate your simulation. So I will suggest that please don't do the simulation like that. Always do the validation with the experiment so that you will know that, I mean, whatever you are doing, it is correct or not. Okay. So now we can start with
yeah. Krishnendu sir, question?
Yes, sure.
So in simulation, does using different sort of machines, does using different kind of hardware, does it have any effect on performing the simulation or not? Because simulation, we are giving all the conditions. So how much freedom do we have to play with the environment, and does the hardware have any effect on it?
Yes, actually the hardware has some effect. So I will suggest that please use whenever you are doing some simulation and comparing yours like one trajectory to another simulated trajectory. I will suggest to have the particular, I mean, the hardware should be similar. And actually, I'm not that developer, but my supervisors and my collaborator, they are like the developer of this Gromos force field, and from there I have heard that you should maintain the similar hardware when you are comparing the results. And the environment is really very I mean, if you are reproducing the one trajectory, I mean, one data, so please give all the similar condition to get I mean your result. But I mean, this is, I mean, still I mean, this is really very like a debate to get the reproduce of the one trajectory, because every time you will do the simulation you will get little bit differences, but you can correlate the data, you can get the RMSD, and also, I mean, the reproducibility of the simulation is, I mean, this is also questionable. Yeah.
Thank you. I guess the right term is the simulation system with GROMACS and other dynamic simulation systems are stochastic in nature. Yeah. And so one should always consider that every time you run the simulation, even though it's on the same machine or on the same supercomputer, there's going to be some variability. So considering the stochastic nature of simulation, one should, plan their experiments and consider their results.
Yeah, I agree with that.
Thank you.
Okay, so now we can start with one simple simulation. Like this is like some protein. This is a COVID protease protein. So first, and we will do the simulation. I mean, we'll have some hands-on on the protein, like only protein simulation in water. Okay, so there are, I mean, many tutorials for this this simulation, and I will suggest to follow this tutorial, because Lemkul is one of the developer of this GROMACS, and they have really the correct I mean, the basic tutorials for all the systems. Like you can see that there is some biophysics system also, which one of our participant asked us in the morning. So you can also have your system like this. So we will follow this EPO system. So you can go to this website here, and you can do all of the simulation, and you can try from here. So I will tell you that this simulation is not the optimized one. So you have to play with your parameters and your the goal of the your simulated proteins, or whatever you are doing. So from this tutorial, you can have some idea about how it is, how the simulation is working, and this is really very basic for the beginner.
So now please, one question from one participant, Hassan. He's asking from yesterday onwards. I think already we explained that we are not aware of that metal containing compounds. Okay, anyhow, how can we create a force field for metal containing compounds, example platinum, and include it in the simulation?
Okay, so as I described, that you have to know the force field first, like which force field you are using. So from there you will get the equation of the force field. So from the equation you will have you will know that what are the parameters you should take to design your force field. So mostly the you have, like I have described earlier, that you have to take the parameters from the experimental data. So from the experimental data you will take the parameters, and you can include this parameter in the force field file. Basically the non-bonded, if you go to the inside the directory of the GROMACS where you have installed it, there you will see the lot of force fields are there. So you have to go to your required force field folder. So in that folder you will see a lot of parameters files are there. So you have to include your parameters inside this this file. So basically you can that way you can design your force field. So like one of my students, he is now working with zinc. So zinc parameters is already exist for inside the GROMACS, but his problem is the protein already have covalent bond with the zinc. So he has to create this, the GROMACS has to know that, okay, this is not the only the metal. So basically the iron, so basically it should not consider it as an ion, but it should consider the bond between the protein, the covalent bond between the protein and with the zinc. So you have to introduce that covalent bond inside the parameters file. So he search the literature and get to know the distance between this bond, these two atoms. So from there he include that distance inside the the bond length, and also he consider the bond angles and all. So in the angle information he include the angles. So in that way he introduced the zinc parameters. So is that clear to you?
Yes, he is asking. Yes, ma'am, Kalini ma'am, continue.
Thank you, Krishnendu. That explains a lot. And Hassan, I am reading your question here. Feels like you are looking for a metal containing compound that you want to, especially platinum, you want to perform the MD simulation. First of all, we never claimed that we are trying to do a MD simulation with metal compounds, but we will be more than happy to help you. The emails are provided of all the instructors. You can email them for your personal requirements, and for docking as well. The session was done. So if you have customized problems, please, ask the tutors on their email. And this session is basically focusing on protein ligand binding simulation, which Krishnendu is taking care of. And if you have customized questions, please do email them. Let's please maintain the decorum of this workshop, because instructors are also human. They are not machines, and they're not, it's not a customized class. I'm sorry, but this kind of negativity will not be tolerated.
Thank you, Kalani ma'am, for your suggestion. And one more participant has asked how to know if we have used NPT or NVT ensemble, and how to put the various parameters such as pH, ionic strength.
Okay, so I mean this thing we will cover later, because I'm just introducing you the.
Okay, you continue, Krishna. Okay.
This protein, the simulation. So we'll go to that session, because it's like very far right now. So now, you have to, know the. Sorry.
I'm sorry, Krishnendu. I'm sorry. I just like to answer one more question. I think the question is from Anil Panwar, and it's about, if we don't have a hardware configuration, could we use paid cloud service? Krishnendu, you could add to this answer. My answer to this would be, there is services by Amazon Cloud and AWS. Well, somebody has answered it, so that's great. So there are paid and non-paid academic licenses from different cloud services that you can get to perform these simulations. As well as, if you are in India, if you're an Indian student, you can always contact CDAC or other supercomputing facility which is in ICMR. You guys can write to them, and being an academician, you get a license for 1 year to use it. Yes, Krishnendu, please continue.
Okay, thank you, Kalani. And okay, so for the simulation, protein simulation, you must have to get the protein. So you will get the structural information of the protein from the PDB database. Like here for the training session, I have used this main protease of COVID. From here, you can download the PDB structure of the protein. So from here, you will have the coordinates of your protein. So then this is suggested that you should exclude the crystallized water, because some protein structure has the crystalline water, and if this water is there, so there is a problem when you will do the simulation, because the force field it doesn't know the way of writing of the crystallized water. So you must follow the atom type of the particular force field you are going to use. So basically the idea is you have to maintain the force field atom types. So you should remove the crystallized water. Instead of that, we will add the force field, this parameterized specialized water for the simulation. And if you have different chain in your protein, and you just want to take one chain, so you should remove the particular chain from there before doing the simulation. Or if you have some monomers, or these dimers or oligomers, and you want to do the simulation of all the oligomers, so there is a command I will show you. You can do that. And just basically you have to clean your protein, and you have to take the desired part of your protein to do the simulation. Okay, so the force field parameters for the protein is already in the GROMACS, and also the force field which you are using basically, and some ions parameters are there. If you have something else, like someone has asked metal, so these parameters you have to design by your own. Or if you have some ligands or something else, so also these parameters you have to design by your own, or there are some many other way. I will show you in the next session when we will do protein ligand simulation. So basically for the protein and like some the 20 amino acid parameters already exist, so you don't have to worry about these parameters. So for today, we don't go for the other things like other ligands or something parameterized. So we will deal with whatever parameters already exist in the GROMACS. Okay, so you have to download the protein, and as I told you, like you have to clean your protein for the simulation. So I have already, for the workshop purpose, I have already clean this protein. So we'll go to that. And, we'll go to the training. Okay. So here already I have the protein, and also I must tell you that if you are running, let's say per day 100 of simulations, so you can make a script and you can run it. Like I will show you my script. Okay, so this is a script of our lab. So here also you can have some sort of script, and you can run it, and you can like minimize your workload. Okay, so in this folder we have the protein. So we will execute our first command, that is here I have also provided this command to all the participant. You can go through this. There is a you can download the files from the, Abraham has sent you some kind of
full drive link. Yes.
Drive link you have sent already, right? So I assume that all the participants have this access to this.
Yes. You can find it in the commands.txt file.
Yeah, from the tx command from txt, so you can find.
Yes, Karan. Karan, he will. Karan is asking about. Can you send us the script that bash script you are showing?
No, I can't send this.
Yeah. Good. Continue.
This is our only. So you can design. It is just a base script, so you can have it. So basically, I will explain the command. So this is like the PDV to GMX command, because you have the PDV file, so the format is different. So you want your, you want that this coordinates should be in the GROMACS format. So for that, there is a command that is called PDV to GMX. So you will execute that command. So I will just. Okay, so I will execute this command. So here you can see there are so many force field you can choose by default. Let's say if you want to use other than the by default by default force field. So let's say you want to use the charm 36M, or like in my project I am basically use. So basically you have to download that particular force field directory, and you have to keep in the same directory as you are working, like the working directory. So from that you will see that the this force field, the new force field will appear here, and from there you can choose the force field. So for this tutorial, actually I am using the Bromos 96 force field. So I will select 14. So it will take the force field. Now I will choose the water model, like what is the water model you want to use. There are several water model available. So if you want to use other than this two water models, so again you have to include your water model in the force field folder. Like let's say you want to use some tip4PD or tip4P or tip3P or whatever. So you have to include that in your directory, and you have to keep the water molecule in your working directory so that it will take it. So for this workshop, I will use SPC water model, the basic one. And now you have you can.
And we guess that all of you have navigated to your respective folder where the files have been stored. Yes, we
Sorry?
guess. I think we guess all the participants are navigated to their respective folder where
Yeah,
these files are staged. Okay, fine. Then you can execute the commands from the terminal.
Yes.
Thank you.
Yes, basically, yes. So here also you can always look your output file. Here you can see the total charge, total mass, and what are the the dihedral things, and what are the everything you can see here. So I will also suggest to keep this one, so you can just really copy all this output, because sometimes you have executed, but for the publication purpose, let's say from after 6 months you will forget everything. So if that someone will ask, like the reviewer will ask, then you will just have nothing in your hand. So for the best practice, what we do, basically we save all the output we create one text file in the current directory, and we keep all the output in that text, so that we will remember everything. Also, you can also copy paste all your commands, whatever you have executed, so that you will remember that, okay, I have execute this command, and you will not forget. Okay, so if you want to know what the command is working and how you will execute, so you can use help, that is flag H, so that you will get to know that what is the command is doing basically. I will just increase. Okay, so the help. So here you will know all the information, like here some description. Here some description about the PDB to GMX. What the flags, there are several flags here. So you will know about your command, and also know about all the flags are doing, and all the description here you will get. And also from everything you will get in the GROMACS website, as well as the GROMACS manual. So please read the GROMACS manual whenever you will start the GROMACS, because GROMACS is really a very big software, and it is really vast, and you should know, and all the the force field information, and all the how this GROMACS is working, and all the theory behind it, it is written over there. So please do read, so that you can clear your things, and you can also ask any expert. Okay, so here we will go through this. So -F, that is flag F, that is for the the input file you will take, and it will give you certain output file. And if you want to name your output, then you can use -O, and it will give you, or by default it will give you conf GRO file, so you don't have to bother about that. And the topology file, how this GROMACS will understand your bond length, bond angle, and all the parameters, things about your protein. So from the topology file, it will, everything is written inside the topology file. I will show you. And this is the this -I is position restraint file. Position restraint is really very important when you are doing the simulation, because, okay, I will tell you this position restraint when we will do the, this equilibration. That time I will tell you the thing, the useness of the position restraint. And as I told you, like if you have oligomers, so you can use merge, so that so that all the chains will be like, there will be like different chains, so it will like treated as one, so that you will have all the oligomers in your file. And if you want to use some certain force field, you can use over here. And also you want to give some certain water molecule other than that by default, so you can give here. And also introduce the salt bridges, like If you have some salt bridges in your protein, you can introduce that. Or if you have some, like if you want to cap your terminals, like whenever you are doing some peptide simulation, that time you must have to cap your terminals. So the N terminal and C terminal you can cap by this TER flags. And you can also define some angle here. And if you have some hydrogen, like some in the X-ray crystallography file, like the protein structure file, if you have some, the hydrogen, like crystallography hydrogen, so that actually creates some mess in the simulation. So you can ignore that, so that we can introduce the parameterized, force field parameterized hydrogens. And if you have some missing, like some amino acids are missing, so you can introduce the missing information in the simulation. But I will suggest don't use that, because it will be really very harmful, and it will create some interprotein interactions. So I will suggest you please model your protein. Let's say you have some gap, like 5 amino acid gap inside your protein. So you can use SPTV, and SPTV will generate 5 amino acid structure, and it will fill the gap, so that you can have the total model. Or you can go for the modeling before simulation, so that you will have complete structure, and you can do the simulation, and that will be really very fine. And rest of the thing you don't have to bother. And if someone has asked, like maintain like pH, so you can also pH you can maintain by the protonation state of your protein from. There are several softwares like website where web server also there, like from there you can know the protonation state of your amino acids, all the amino acid in your protein. So in your force field parameters you can introduce this protonation state, and by which you can maintain the pH. Okay, so I think this much is clear. And here you can see that we have generated the GRO file. I will open the GRO file and show you how so this is the GROMACS way of writing the coordinates. So here, this is the information of all atoms. Like these numbers of atoms are inside this file. So you can also make GRO file by your own. So like this is the number. Here is the the amino acids, the atom types. Like in the PDB format, the atom types are maybe different, but this is the way of GROMACS write the atom types. And this is the numbers, how many numbers of atom, and this is the coordinate. Always remember that the X-ray crystallography units are in angstrom, but in GROMACS everything is nanometer. I don't know why, because maybe the developer like nanometers. So all the things, also you can see the units of whatever units are using in the GROMACS in the manual. So please do have a look, because these are really very important. So this is the only difference between the X-ray crystallography file, like the PDB file, and the GROMACS file. So these are in the nanometer. Always remember that. And this is the box information basically. Okay, so I will show you the topology file. Okay, so these are the topology file where you can know the executed command we have executed here, and the force field, like which force field we have used. Here you can see the all information about the all the amino acids in the protein. And here the charge is defined by GROMACS and the mass. And okay, and some bond information like the bond length and the bond the bond angles, like I will show you maybe. And the dihedral, mostly it is written over here. So this is really very big file. Okay, so bear with me, please. I'll show you. So this, I think this is the dihedral information, and this is the angle information. I guess so. I have to check. And the bond length. Yeah, this is the bond length information. Okay, so here. Yeah, this is the bond length information. Then you have the dihedral information and the bond angle. So these are the basically three parameters GROMACS write in the topology, as well as this is the position resistant, I will tell you later, and this is the water model, and this is the force field parameters for the ions. So when you are using something else, so you can write over here. Let's say in your simulation you have some ionic liquid. So for that you can go here. You can go here and write has include and the ITP file, like the ionic liquid file information. So you have to design the ITP file before, and you can give the information over here so that it can include the ionic liquid as well, along with the water. So anything you can like play with it, and you can develop your own parameters. And these are the molecular information, like now protein chain A, it is like one molecule it has taken. Okay, so now we can go to the next command. So now we will create one box. Like just imagine our protein structure is there, and now we will create one virtual box along with the protein. It will keep the protein inside, and we are creating one virtual box. So for that, this is the command I will show you. This is the edit command, and it will take the input from the previous output, like the conf.gro. This is the coordinate file of the protein, and this is the output. Like I generated this output file new box, and this is the center. Actually, I just force my protein to inside the center of the box. And this is the dimension of, this is the, not the dimension. It will create, it will just keep the protein, and from the outer space of the protein, it will just generate the box like 1.5 nanometer distance. So these are the distance we have maintained from outside the box, and this is the box type. So you can use different types of box, but actually people basically use cubic or there are certain kind of box, but it's based on what is the size of your protein and how the protein is look like. If your protein is rod shape, then you should use triclinic box, so that it will accommodate and it will not generate unwanted gap. So based on the protein, how it look like, based on that you can design your box. So our protein is like something globular protein, so I prefer the cubic box here. So again, you can know the information of the command as I told you by help. So these are the help. You can get to know your, the command, how to use the command. You can use different other flags to modify and
hello.
Abhishek, do you have any doubt?
Am I audible?
Yeah. You raise your hand before you ask the question, Abhishek. Okay, fine. Go ahead.
So, sir, as I already asked that how to use, when to know we have to use NVT or NPT ensemble.
Please wait, because we have not reached that much, because I have just started. Please bear with me, because these are the things you are asking about the equilibration. So please wait. I will cover with that. So please.
Yeah. Sir, I just want to say something else also. Yeah. So, sir, it would be better that if you give some theoretical aspects also, like what are the various cases and how to tackle them. Like I am asking NVT and NPT ensemble. Maybe there are many things. Just to give some gist. I am not saying go into each and every detail. That is not possible. But anyhow,
so what exactly you want me to cover with? Because actually we have also the time constraint. So I have,
sir,
what exactly you want to know? Because I don't know. You are saying that okay, I must.
Yes, you can carry on, Pratik.
Okay, sir, no problem. You can carry on.
Pratik, Dr. Pratik.
Okay, so as I told you, like there are different kind of box you can use based on your protein information. So here you can see that the box information also they are writing cubic and octahedron and whatever you can use based on your protein. How it looks and how it's like, and many things you can cover over here. So these are the information you should know, like you should know your commands. So I will suggest that please go to the manual and know each and all the commands, whatever they are doing, so that that will be like very much easier to use GROMACS. Because I can assume that you maybe for the beginner it is hard to understand, but whenever you will used to it will be like very easy. Okay, so now we have created the box outside of the protein. So this is my file, and the updated file. This is initially we have generated this conf growth. Now you have like new box file. So now we will put water inside the box. So for that you will use Solvet command. Okay, so this you can see, as I told you before, that please always save this information, because these are really very important. You can see here the output box content, the volume density. Maybe sometime the reviewer may ask, so it's really good practice to save it. And this number of water molecule inside, it entered inside the box. Okay, so now we will understand what is the command. So basically you are giving the input by CP, the new box in, this is the box, the output from the previous command, and you are giving the which kind of water model it will include inside your box. So this file is already exist in the GROMACS folder. You can see if you go to the GROMACS folder, inside the GROMACS folder you will find this water model. So, and also I will recommend all the participants that you should go inside the GROMACS folder and just play with it and get to know that what are the information is there, because this is not a black box. So everything is open, and you can just enter into the directory, and you can just know that what is the parameters GROMACS is using, and you can play with that. So that will be very useful when you are working with something new, I mean, new things other than whatever in parameters are encoded. Okay, so this is the output, and this is the topology we have used. So now it's generated the solvated file, and also it modified this topology. We'll see. So see here the water molecule has been added, like this much of water molecule we have added. Okay, so now actually sometime we have the common error that the topology every time modified. Let's say you are let's say you are in this solvation step and you have some error, and already in the topology file the water has been modified, and you want to go back and you see that okay, there is some error, and you will just correct it. So, and you just give the command of the previous command, and you solve the error. But always remember to update your topology as well, because if you have shown that in each and every step the topology is modified. So if you have added this number of water molecule, so the topology is not modified, then again you will add extra water in the topology file. Maybe in the GRO file, this GRO file, this much number of water is modified, but here you have forgotten that your topology is not modified. So always check your GRO and solvation, this topology file, so that you can rectify your error. So I will show you how the boxes look like. Actually, it is always very good practice to check the GRO file in each of step, because actually I cannot show you because I have logged into the supercomputer, so that's why I can't show you, but I have downloaded and turned into PDB file. Actually, you can use BMD. There is another program for the visualization. You can use BMD to just BMD command and config this whatever GRO file you want to see, and enter. You will see visualize the this code, this GRO file, your protein file and whatever solvated and all. But as I told you, like BMD will not work in the GUI version, so I have already downloaded. I will show you how our system look like. So I have converted this GRO to PDB, and here we can see that how our system is look like. So here you can see we have generated one box. So just imagine you have the cell is outside, and this protein is cytosolic protein. So we have just cut a chunk of the cell from your cell. So basically this is the protein here inside the box, and you have just created one virtual box like this, and inside the box you have filled the water molecule. So these are the water molecule you have filled, and this is the protein. Okay. So now we will add the ions, because the cell in the cellular condition there is some ions, and it has been shown that this ion concentration is always like approximately I think 150 millimolar. So you have to maintain the concentration. So if you have some specialized, you are doing some specialized simulation and you have like experimental list has given you certain, the concentration that, okay, so my system will be like 50 millimolar. So you can maintain that 50 millimolar concentration in your system. So basically, generally when you you have no idea about the concentration, so please always include 150 millimolar concentration of the system. Okay, so we are here. So we will add the ions. So for adding the ions, this is the gen ion command. So the gen ion commands here. Okay, actually there's something wrong. I have not. Okay, so sorry. Okay, so you have to create one TPR file first. The TPR file contains all the force field parameters. So sorry, actually I just skip one command. Okay. So now we will keep. Okay, there is some error. Okay, so always very good practice to know the error, like what are the error is coming. So here I have not given the parameters file for the adding of the ions, so that this errors is coming. So now you have to keep your parameters file inside your working directory, so that this type of error will not come. So when you are doing the simulation, like especially in the GROMACS, you will face lot of errors. So that how to tackle that error, that is really very interesting. Like these are like error. From the errors you will get to know like how to do the simulation correctly. And please read all the information. Like here it is clearly showing that this MDP file is not there, or it is not accessible, so that this type of error is coming. So always GROMACS will give some clue that what is the error and how to tackle that error. So now you can see that in our directory there is no MDP file. So actually I have prepared all the MDP, that is parameters file, I have prepared, and I have sent all the parameters file to all the participants. You can download from there. Okay, so okay, so I will tell you what this command is doing. That is the copy command, and all the parameters are in my previous directory. So this is star, means it is copying all the files with naming and ending with MDP. And this dot is saying that, okay, this copy will be to this particular directory, that working directory. So see, I have all the parameters file over here. So now I can execute the command. Okay, so it generated the force field file, the TPR1, and now we can execute our ion addition command, that is this one. Okay, so now I will tell you what is exactly it is doing. So basically I am adding Na and Cl, and this this file, this flag is saying that it will neutral the system, and this is the concentration we want. Again, I will tell you, you have to know what are the unit GROMACS is using. So here is 1.5 is the, this is the millimolar, like 150 millimolar. Okay, so you will add your ions in outside of the, inside of the box, and you will just, this command will replace all like the water molecule and place the NaCl ions inside your system. So for that we will select water molecule, then enter. So here you can see it is replacing the solvent like the water, and you can also see the number, the atom number over here. The atom number over here, and the number of the water molecule. The ID of the water molecule you can see, and it replaced with the Cl, and also Na also replaced with some other water molecule. So now we can see the topology file. See, this number of the ions are added. So it neutralizes the system. If you remember that our system is, previously our system was -4 charge. So that's why it compensate with the, it neutralize the system, and this number of ions has been added to maintain the concentration of the system. So here is the file with which is like solvated as well as ion is added. So we can have a look how it looks like. So this is the
Karan is saying some error that the constraint file is not available. I think he has to modify that topology file.
Mm-hmm. Sorry?
Karan is saying that he found an error that constraint file is not available while he's typing the previous command.
Which file? I can't hear you.
Constraint file. Yeah, Karan, go ahead.
Hello, sir. Sir, I got an error that is saying that cannot find a position restraint file, restraint.grow.
Okay,
while
so
running the next previous command.
So have you run the PDB2 GROMACS, PDB2 GMX command?
Yes, sir.
So is your folder is containing this file, this I2P file?
Oh, yeah, yes, sir. That is there, but that is quoted with H.
Okay, so
that file is there, sir. That file is there.
Okay, just rename. Okay, so actually, what is the problem? Like these are some file, like these are like modified file. Here you can see in my directory also, like these are like this modified file. I told you, like every time you will run any command, it will modify the topology. Similarly, your actually I have given you this file already. So maybe it is like updated, so you just rename it as it is, like PO SRE.ITP, so that the command will be, it will just take this file, just rename it, use the MB command. Okay, so we can have a visualize how it look like. Okay, here you can see that we have added the ions. Like there is protein, there is water, and ions are added. So now we will, our system is ready, and we will go for the minimization. So again, you have to generate the TPR file. And you have to run the minimization. For the running, you can use the MD run command, so it will just run. Okay. So while running, if any participant has any question, it will take some time.
Sir, can I see that previous command, sir? Command list that you were showing?
Actually, this command list already you have.
Prateek sir has addressed something about NVT and NPT as an ensemble.
Yes, yeah, actually, so the people were asking about NVT and NPT. So what happens is that after energy minimization, so yeah, I have actually also replied in the chat box also. So the energy minimization happens at 0 Kelvin, and then the simulation starts at a finite temperature, which is around 310 Kelvin, or in other systems of interest. So there is an abrupt increase in temperature because of the same. So to stabilize the dynamicity of the molecules, initially a constant volume temperature is maintained, so that there is, if constant volume is maintained, there will be no PV, no work. P into V is the work function. No work will be attained. So there, the temperature which is given will be initially used to dynamize or mobilize the molecular motions, and then followed by the NPT simulation, constant pressure, constant temperature simulation. The other thing is that people may do, there are like many groups which are doing simulated annealing just after the minimization, which is increase the temperature gradually from 1 Kelvin to whatever Kelvin you need, 300, 310 Kelvin, etc. Gradually simulated annealing at constant volume conditions, NVT, canonical ensemble, and then you follow it by the NPT. Why we should follow by NPT is because in any kind of biomolecular system can actually fine-tune their volume, because the normal conditions, ambient conditions, are constant temperature and pressure, and there is a change in volume. But nevertheless, as I had discussed in the previous session, is that for proteins the PV term is quite less, maybe around a few kcal per mole. So if the system is immediately running stably with NPT simulation, then you can continue doing the NPT simulation from the beginning itself, and can actually ignore first few nanosecond of simulations for analysis.
Yes, that was pretty clear, Prateek. Thanks for your.
Okay. Thank you.
Thanks, Prateek, for.
Thank you.
Answer.
Actually, yeah, one participant would like to ask some. Anil Panwar, can you ask your question?
Yes, sir. Yes, sir. My question is that when we open this EM file, and when we open this EM file, energy minimization file, and there is Fmax less than 1000, then what is this less than 1000? What did it mean?
Okay. Actually, it is running right now. Okay, so what exactly we're asking, that it is less. Sorry, can you please repeat the question?
My question is that the force, maximum force, is less than 1000, like that. In this file, EM file.
You mean this file?
Yes, you can open MDP file. Yes, you open this file, and there is.
I think here there should not be anything like that.
Maximum force, there is. I can't see this.
Actually, I am using this.
I think it's not the default file we get from the tutorial.
Which tutorial?
Lysozymes water.
Okay.
I think you alter this somewhere else.
Yeah, actually, the tutorial file is not the optimum, so that's why I have given you I mean what exactly we are using that file. And what you are asking, I think they want to, you know, some cutoff they want to give for the force, and they doesn't want that it should go beyond that cutoff.
Yes, sir.
Yeah. So I will suggest that please don't follow everything from the tutorials, because this is just for the beginner. So please use, and also these parameters and everything is given in the manual. So please do go to the manual and I mean check it, so that you will clear the doubt by yourself. So, okay, so now
yes, continue.
Like it's done. So here we have generated the EMGRO file, which we will. This is the energy minimized file. So yesterday also one.
Sir, I can stop you.
Yes,
Yeah.
yes.
Yes, you can see in your results. Steepest descent converted Fmax less than 500 in 2,301 steps. In your file,
This is the, I mean, they want to control it. So if you want to control the minimization, so you can use that flag. So there is no issue.
So how many steps it takes to converge?
It depends on the protein.
Yes.
It's like there is no such I mean step. So here is we have given that if I will open, we have given that it will run for 50,000 steps. So you can increase that step. So there is no problem. You can modify all the these parameters as per your simulation. So this is the thing, you can control your simulation. So you can add, like here we are using steepest descent. So this is not only the algorithm is there, so there is also conjugate gradient, so you can use that as well. So it's like fully up to you how you are going to do your simulation and how you want to achieve the result. So it is based on the customization, so you can use this. So please go to the manual and do have a look that what exactly these parameters are doing and how you can control your system, because this tutorial, what I am showing you, or whatever this LAMCOOL has shown in their tutorial, so these are not the optimum one.
Yes, Krishna, continue.
Okay, so now we have minimized the systems. Like yesterday, one question was there, like how we can minimize the system. So this is the way you can minimize your system. So here we have minimized our system after the solvation. So you can also minimize your system in the gas state, like without solvation. Okay, so now we can do the NVT, like your system is equilibrated. So now energy minimized. So now you equilibrate your system. So just imagine that you wanted a glass of ink solution. So you will pour the water. You will take the glass of water and you will pour 2 drops of ink inside the glass. So it will take a lot of time to get the proper solution. So you have to stir the glass with some glass rod so that it will be properly equilibrated. So here also we want that our system should be properly equilibrated, because we have this now. This is completely static. Like your protein is static, the inside box, the water ions, everything is static. So you want to give some dynamics, and you want to give some some certain parameters, like as I told you, like pressure and temperature we will add to mimic the environment. So I'm not going to explaining all the theory, because already theory has been explained. So I am trying to give as much as possible in a layman way. So here basically we will go to the NVT NVT ensemble first, and then the NPT. So here, as one question was asked, and thanks to Dr. Pratit, he has answered that. So actually I wanted to explain here. So here basically what you are doing, you are just adding the temperature. Okay, so you are adding the temperature. So here in the tutorial purpose, I am just doing the, okay, I will show you the parameters file, NVT. Here, I am giving the temperature like 300 Kelvin. Always this is in Kelvin. So please remember all this thing, or you can just go to the manual and check the what exactly the unit of all the units the GROMACS is using. So this is the temperature for the system we want to achieve, and we want to do the simulation. So it is not recommended when you are doing some kind of specialized simulation, let's say DNA simulation you are doing. So you should not suddenly increase the temperature like up to 310. So you should increase the temperature gradually, like let's say 50 Kelvin, then 100 Kelvin, like that. So you can make multiple MDP file, and you can run like let's say 10 picosecond simulation. Here you can control the time. So let's say run 10 picosecond simulation for 50 Kelvin, then again that output you can use for the next NVT, so that gradually you should increase the temperature for the like some specialized simulation, so that you should not break the system. So just imagine that you are warming some water. So when you are warming the temperature, you want to achieve the temperature of the system in the water to like 310 Kelvin, but it cannot achieve like suddenly. So it will increase gradually. So just feel the meaning of the gradual temperature increasing. So like that you also gradually increase the temperature, so that you will not disturb your system. So with the sudden.
Aditi would like to ask one question. Hello, Aditi. Okay, go ahead.
Okay, so another thing I want to say, the position restraint. So when you are also doing the simulation, at that time you should position restraint. Restraint itself is saying position means the position of the atom, and restraint is you are giving some force to the atom. So just imagine everything is static in right now. So you just don't want to disturb your system like with sudden shock, so that you will restrain all the atoms, like these are the atom information, and these are the coordinates like X, Y, Z coordinates. So you are giving the 1000 I forgot the units, so please do have a look in the manual. So you are applying the 1000 this unit, I forgot, force to the atom, so that it will not move. So your protein will be in static, and the environment like water and everything will move, so that you will not disturb your protein. So when you are gradually increasing the this temperature, as I told earlier, you are also gradually release the this position restraint. Like now you put it 1000, so you can release like 800 or something lower. So you should decrease the position restraint, so that your protein will not be disturbed, or your system like your like DNA. Basically, when you are doing the DNA simulation, this is really very important because DNA is not stable like protein, so it will just collapse. So you should maintain the the position restraint very carefully at that time. So like that, you are making a lot of so many MDP file. You should create so many ITP, position restraint file, so that you can define, like here every attempt is defined. So you can define like if you want to restrain your all the hydrogen or all the side chain or specific like some binding site you want to restrain with your ligands, or some, let's say the backbone you want to restrain. So you can restrain everything here in this file. For the restraining, there is another command called, I think, Genstar or something like that. I have to Google it. There is another command from where you can create this file. Yeah. Sorry. I forgot actually.
Yes, Anil has some question. Just Anil, can you ask?
Yes, sir. My question is that when we talk about the MD simulation, time of MD simulation, we talk about the final command MD run, like that 1 nanosecond to 10 nanosecond.
MD run you are talking about.
Yes, yes.
Yes, you will come to that MD run.
Yes. My question is that what if there is no time limit to NVT and NPT in these simulations time? Means like that we run for 10 picosecond or 100 second cutoff for that.
No, you can run it like whatever you like, because actually here you want to just give the system feel like some sort of like equilibrated. So this is not like optimum step. So for the tutorial purpose, actually I am just giving you example that okay, you can run here I am running like 100 picoseconds. So you can run up to 1 nanosecond to 2 nanosecond to achieve the equilibration. So you will check the density of the system, or you will check the energy of the system while you are running. And also here in the tutorial, they have suggested like you should check the temperature, and you should get a linear curve like here.
Does this equilibration time depend upon the size of the protein?
It not really. I mean, it should be like you will see the temperature variant, because the NVT were basically talking about the temperature. So here you can see this is really very fluctuated, and they have only done 100 picosecond. So you want something like straight curve, so that you will know that okay, this is properly equilibrated. Maybe Dr. Pratit can add something.
Yes, Krishna, you can continue, Krishna.
Okay, so basically you will check that this particularly the temperature. So for that, for this getting the temperature, you can use GMX energy command to and along with the EDR file, you can generate the temperature. So and you have to give this. Just remember this 16 and 0. So it will give you the temperature curve, and from there you can get to know. So there is no optimum like optimize rule. So based on your system, your whatever you are doing, so based on that you have to select the time scale. Okay. So now we can do the, there is some error in. something is showing that the error is user input is C, and this is it doesn't have the.
Okay, Krishna. Here you have to change the command. Actually, with the NVT parameter, you have to give the em.
Okay. This is like GRO. Sorry.
Yeah.
Sorry, em.gro. I'm sorry.
Yeah. Oh.
Yeah. Actually, I have just copied from my old script, and there we basically do lot of so many NVT run. So I just use the previous one. So here basically you have to use the input file, sorry, the output file from the previous commands. So thank you, Rohit. So, okay, so as I told you, like GROMACS is really very friendly here. It can tell you like what are the error, and it can give you some clues. So from there you can solve the error. And also there is GROMACS community. You can also mail them, and they are really very friendly, and anyone can reply you. Okay, so now we can run. This command gmx one. I want to see how to run in multiple. I have actually forgot how. I think this is Rohit. Can you please help me with how to run in multi-thread, like using multiple?
Yes.
This is like N, right?
Yeah, NT,
right?
Hyphen NT, yeah.
Okay.
You can use hyphen NT. And, you can discuss the code. And your last part of terminal is not visible actually, Krishna. So your last part of the terminal where you are typing the command, that is not visible in this.
Okay,
Maybe
so
now it's visible. Yeah.
Okay. So I have to use NT. Let's say 4. So NT4 is like you can run your script with command with more number of processors. So you will use the NT. So it will run in multiple processor. Okay, so maybe we can stop here because I think I am crossing the time limit. So
anyhow, we'll be showing this analysis of the results in next session. From 3 to 5. Yeah, anyhow, we have to resume something, and we'll give break to the participants also. They'll have their lunch.
Yeah, sure.
Meanwhile, we'll giving the recording sessions. They'll practice anyhow.
Yeah.
Thank you, Krishna.
And I will show you after. This will take a long time, so I will show you the result. This is only the output file, GRO file you will get, and we will use for the pressure. We will add pressure like in NTP.
Yes, sir. We'll happy to accept the questions in the afternoon session. Karan Kumar has asked one question that will be addressed in the afternoon session. Is it fine, Krishna?
Yeah.
Session now.
Sure,
thank you.
Sure. Okay.
We again meet by 3 p.m. Please be ready, all the participants. Thank you for your patience. I'm closing the session now.
Thank you.